Hello, everybody. Good morning. We will be starting the webinar here shortly. We'll just give it a few more minutes, please. Feel free to introduce yourselves on the chat box. And as we go through the session today, feel free also to post your questions, and we'll be happy to answer them as we go through the session. Welcome, everybody. Just want to take this opportunity before we start. We have Anushka Jain, biotech graduate. Also, we have Robert Weinberg, welcome. Salsabil Idris, welcome to the webinar. And we also have Eric Smith. Welcome to the webinar. Feel free to introduce yourself on the chat box. Sonalika, can we go ahead and start? Okay. Good morning, everyone. Do you hear me all right? Loud and clear? Just want to make sure everything's clear on my end.
We hear you great.
All right, perfect. Thank you. My name is Japs. I'm one of the marketing specialists here at Pine Biotech, working for the OmicsLogic portal, or programs rather, and I would like to welcome to this overview session on the summer 2022 OmicsLogic virtual training programs. The session will give you additional details about the program registration, levels of complexity, and desired outcomes you might be able to achieve over the summer. Now, we have a range of options that you can look into further and register by the end of the second week of June. The programs will be offered virtually around the topics of coding in R and Python, infectious and chronic diseases, as well as a research project program that will help you complete a research project leveraging public domain data, big data infrastructure, training, and support from our mentors. Now, because ultimately the methods are best learned in the context of real case studies prepared by our network of mentors and collaborators that illustrate how various bioinformatics methods and omics data types are used in current research in medicine, biotechnology, agriculture, and even astrobiology. The projects are prepared by taking publicly available data sets and using them to explore and understand both the research challenge and the analysis path to extract meaningful insights from complex omics data. The OmicsLogic portal gives the students options for analyzing a given data set, code-dependent paths or code-independent paths. Now, these data sets and associated publications can be used in several ways. You can learn bioinformatics without any coding at all. For that, in each project, we explain the analysis pipelines on the user-friendly TBio platform for big data analysis. You can also learn the scripts behind many of the steps, learning to wrangle, clean, visualize, and analyze data in popular languages like R and Python in order to create your own analysis. Finally, you can hear about the projects and their research applications in our mentor-guided programs where experts share their experience and support research projects developed by you. Let's start by signing up to learn OmicsLogic portal. And this educational portal will help you get started on your bioinformatics journey. We will be posting the link on the chat box, and we will give you a few minutes to visit the OmicsLogic portal and be able to sign up if you still have not created your profile. I would like to encourage everyone, for those who have not created their profile, this will be a great time for you to be able to get started. And if you have any questions or issues in signing up, please feel free to reach out and one of our team will be able to assist you. So the link is learn.omicslogic.com. And let me know if you have trouble looking for the page where you need to sign up. It's at the top right of the page. So for those who have signed up on the portal, if you can please type yes in the chat box. Okay, perfect. We have a couple people who already have signed up. Anybody having trouble signing up on the OmicsLogic portal right now? Yes, the link is learn.omicslogic.com. I'm posting that link right there. Okay, thank you, Robert. All right. Okay, so I'm going to move on. So when you create your profile, we would like to encourage you to update your profile in order to track activity and issue achievement badges and certifications. Now, a complete profile includes a profile picture. You can use your avatar or a university logo if you don't want to use your personal picture. You may also connect a social media account, either Facebook, LinkedIn, Instagram, or Twitter account. And last but not the least is your biography, in which if you can please provide a short or brief bio about yourself, including your research interests. So this is what you will see when you have completed when you sign up for the portal. And this is where you will see your activity. A couple of tabs that I want to introduce is the activity tab, the courses tab, programs, and certificates tab. So as you complete or enroll courses, this is where you will actually see all the activities, and we will get through that on the next slides. So after successfully completion of a program, you will be awarded with a certificate, and you'll be able to view or access your certifications through your profile. If you see earlier on the previous slide, there is a certificate tab. That's where you can access all the completed certifications, and you'll be able to download it from there as well. After you register, you will be redirected to the courses tab. And here you can search for any course title and use the tags to find series of resources around a topic, or choose one of the collections down below, which is highlighted in red. And in this video, we will look at the Getting Started collection. I also would like to add that these Getting Started courses are free and for anyone who wants to get into the bioinformatics, and you're welcome to share with your network as well to take these courses. Under the Programs tab of your profile is where you find the assigned programs of your choice. Now, each program has a schedule, sequence of events, and every event has recorded sessions and assignments designed around that topic. And each activity you complete will get points, which are for completing the course, which is 500 points, running a pipeline, 100 points. As soon as you update your profile, you also get another 100 points. And for any group activity that you complete is about 10 points. And you will see that under the activity tab with all the logs or every mileage that you have completed. Now, in this program, you will also be taken through project examples on the OmicsLogic portal, which will help you understand the practical applications of various omics data analysis and learn how it will be applied to answer research questions in hand, and finally designing your own bioinformatics research project, indeed. And during this meeting, you'll be able to learn about the upcoming training programs offered by the National Association of Idea Principal Investigators, or NEPI, on bioinformatics. We will focus on 3 programs: Bioinformatics for Infectious Diseases, Genomics and Transcriptomic data analysis to study viral and bacterial pathogens, as well as host immune responses, Transcriptomic data analysis for chronic diseases such as cancer, metabolic, and many others, Bioinformatics using R and Python, getting started with sequence and structured data analysis in R or Python. In addition, we will be sharing more information about the OmicsLogic Research Fellowship, a program designed to help young investigators complete a full bioinformatics project over the summer and present it during our virtual summer research symposium. To achieve this goal, we use a combination of online sessions, self-guided study materials, and practical assignments for an immersive experience that has proven to be effective in our well-known and respected OmicsLogic programs. Now, to talk in detail about the upcoming summer programs, I would like to invite Dr. Raghavendran. Dr. Raghavendran is an accomplished computational biologist with experience in large-scale analysis of multi-omics data and data science methods in precision oncology and protein DNA structural bioinformatics. I will pass on the stage to you, Dr. Raghavendran.
Thank you very much, Jabs. Let me find ways to share my screen and confirm that I am audible to all of you.
Sure.
Can you stop sharing your screen, Jabs?
Sure. Bear with me.
Mm-hmm.
Okay. There you go. Thank you.
Thank you very much. And please confirm that you can see my screen.
Yes, Dr. Raghavendran, we can see your screen and your audio.
Perfect. Thank you very much. Okay. So I thank once again Jabs, and I'm Raghavendran, bioinformatics mentor with Pine Biotech. And I welcome you all to this overview session on the Summer 22 OmicsLogic training programs. So this part of the session will give you more detail and additional details about the program design, about the curriculum, and about resources. So as you know, this summer we have a range of programs that you can look into further and register so that you can learn a little bit more about bioinformatics and how it is applied to various different topics related to biology and bioinformatics. So this program will generally be offered virtually around the topics of infectious diseases. It also includes coding in R and in Python. It also includes bioinformatics analysis of gene expression data in transcriptomics, and in general, introduction to big data bioinformatics. So this program will actually help you to complete a research. At the end of this, we also have a research fellowship program, and Dr. Mohit will talk more about this research fellowship program. So this research fellowship program is specifically designed to help you complete a project leveraging public domain data from using big data infrastructure and exploiting training and support from all of us, from our team and our mentors. So let's go to a little bit more detail about these programs one by one and understand what they offer to students and faculties. So we start with introduction to bioinformatics, where we actually introduce you to the big world of bioinformatics and big data.
Sorry for interruption. Hello. Can you hear me?
Sonalika, can you mute? Yes, thank you very much.
Yes, sir, you can proceed further. Shivam, can you put your query in the chat, please? We'll answer your query towards the end of the session. Thank you so much. Over to you, sir.
Yes, thank you very much. Please, please put your queries and enter your queries in the chat. I'm following chat as I'm chatting with you. So, so let me share again. Okay. So the Introduction to Bioinformatics program is designed to introduce you to the world of big data biology and data science tools. So it starts with the basics of next generation sequencing, and it starts with what are the different omics data types that you will be learning, and what are the different omics data types that are used generally to understand the biology and different biology of the system from molecular level. It also extends to what are the analysis methods and options that we have to use for or to employ on different omics data types, so that we can extract the maximum information from these data types. Okay, somebody says that my voice is so feeble. Let me sound up. Thank you very much. So in the end, by the end of this big data bioinformatics program, you'll also get an introduction to how to use or how to apply advanced statistical methods like machine learning and deep learning to the structured data that we generate from this next generation sequencing data. So in the end, you will achieve all of your achievements will be summarized in certificates that you can share with your peers. So yeah. This you went through. We have several extensive training materials prepared for you, like associated courses, lessons from OmicsLogic platform, asynchronous videos, live hands-on demo sessions, and we also have prepared curated example projects and which takes a publication and the data associated with the publication to an analysis method of its own and walks you through the whole process of from the data set through through specifically designed analysis methods for the data set and biological interpretation and conclusion and comparison with the original findings. And we also have in the research fellowship program that you will hear today. So we promote project-based learning and interactive sessions for exploring bioinformatics as an option. So we also have several lessons which are coding lessons that that is structured in an interactive fashion so that you can explore coding for bioinformatics. So the analysis methods that we have discussed or that we have in hand for different omics data are usually applied or employed in two approaches. So one of the approach is for code independent approach is for those who does not want to venture into the world of coding and who is not very comfortable in coding. So these methods, these code independent infrastructure methods will include hands-on exercises in our big data bioinfo platform like Bioinfo platform. And this provides a simple and intuitive user interface for processing, analyzing and digital data and different structured data also. So we can employ all different kinds of analysis methods. Here you will get a 5 terabyte storage so that you can store your data that usually is huge when you involve big data bioinformatics, and you will get support from us, and you'll also get all the necessary associated resources and lessons and videos so that you can set up your analysis pipeline independently and vary the methods in the analysis pipeline to suit your data. So this analysis pipelines include both the analysis of next generation sequencing data to arrive at a structured table, to structured data or a structured list of variants. And then from there, it also includes the downstream analysis of whatever you have generated, the structured data that you have generated, so that the downstream analysis applies various statistical methods, machine learning methods, so that you can infer and explore the meaningful relationships that is buried deep within your data. So here is an example of the code independent pipeline that I was talking about, and this pipeline builder interface in the Bioinfo platform is color coded so that you can establish a connection between a logical connection between how your analysis methods are progressing. And the data input is an input file, and the analysis can be chosen based on the type of research and the type of question that you are going to ask. We usually have 3 basic colors that separates the analysis methods. Blue is for pre-processing, red is for mapping, and green is for quantification. And you can explore orange is for downstream analysis. The interactive output, we also have option for interactive output, which allows for easy visualization of the data outputs. And there are several demo pipelines which will give you an experience of setting up an original pipeline and downloading the output, associated output, to get a feel for how to set up big data bioinfo informatics pipeline for next generation sequencing data and for downstream analysis, and what are the different kind of outputs that you will get and how you can make sense of this output in the connotation with the biological relevance or phenotypical association of the samples. Yeah, so this is part of the interactive output. So for those who want to expand your skill sets to coding and venture into learning coding along with their analysis, we have lessons that involve coding, and these lessons are divided into several steps. So the first step is always to understand the context or the aim of the analysis, and it is followed with the logic behind the analysis that we are performing, and then we have a learn the code section. Learn the code section, where each and every parts of this code are explained to you in a small piece by piece manner. And finally, you'll have practice the code section, where you can practice whatever you have learned. And then in the end, you'll have option to download, or you'll have option to access the full code document in a Jupyter notebook or a Google Colab notebook format, so that you can independently modify the code to suit your analysis or data requirements. So each session is actually planned for an hour-long video, and you will have more associated resources that you will have to go through. And I think our Sonalika will actually talk more about how much you should spend for each and every one of these sessions, so that you can gain or get maximum information from these sessions or resources from these sessions. So yeah. So for our next program, we have a biomedical data analysis using R and Python. So the introduction to bioinformatics section will introduce you to the different analysis methods. But if you want to delve deep into the world of data analysis and data science of biomics data or big omics data, then you have to choose an option like this. So in this program, for example, the focus is on learning data science and not programming. So programming we use as a tool to learn data science. The more focus and emphasis is on understanding the logic behind the methods and tweaking certain part of the programming or of the code blocks, so that it suits your data or your analysis methods. So yeah. So why do we choose only Python and R? So we know that Python and R are the major resources that is used for bioinformatics data analysis. And I don't want to create any war between people who like Python and people who like R. So we are on the fence, or we are of an opinion that it is not a good idea as a bioinformatics enthusiast or of a data science enthusiast to pigeonhole yourself into one of these methods. Both have their advantage and both have their shortcomings. So you should learn enough concepts from both the tools as much as you can, so that you can apply either one of them depending upon the type of data, depending upon the type of analysis that you use. And that is what we also promote. For most of the lessons that you have, we have all these analysis methods divided into 3 parts. The first part, as I actually mentioned earlier, it's code independent block, where you can get the experience of performing these analysis using the big data bioinfo platform. And the second portion is running the same analysis in an environment like R. And the third one is using environment like Python. So you will have complementary information from all these 3 different resources. So we first start with the basics of getting started with R and Python, where we start absolutely from the basic, like how to load a data into R's environment, or how to load a file into Python's environment, which is absolutely the most necessary skill that you want to have if you want to use R and Python for bioinformatics data analysis. And then we up the ante with trying to analyze or trying to read DNA sequences, and how to compare DNA sequences in R and Python, and how to take your first steps into bioinformatics data analysis. So all of the data that is discussed and all of the data that you are going to load are from either of the omics data, specifically prepared and specially prepared for you to run all these analysis in your in the interactive platform that you will learn, or in your R studio that you have locally installed, or Python environment that you have locally installed in your computer. And we and then to increase the applications of R and Python into the world of data science. We have several different data processing and visualization and advanced visualization methods that you will get hands-on to, and how different visualization is also going to help you learn about the patterns that you can generally observe from the data. It's just not you run this code, boom, you get a scatter plot. If you run this code, you get a histogram, but more emphasis is on what are you going to learn from histogram, and why is histogram a better option when compared to scatter plot. So more of these kind of discussion is what we will be having in this one. So to start from absolute basic, you always have to start from somewhere. So we have to give you a detailed introduction in the first lesson, or introduction to bioinformatic analysis in R, about some of the concepts that you will mostly use, or you will use frequently in your data analysis, like what are the different objects like data frame and the array and matrix that you will be using for your omics data analysis. What are the different basic functions, like how to find the dimension of the array, how to find the size of some of the elements, and how to find the how to index or take specific portions from the data frame, or how to restructure data frame based on your requirements. So all of these would be covered extensively up at the start, and then as you progress in the program, so these will be used repetitively, so that you can get a feeling for how these are used in the omics data analysis. So you can say that we in Python or in our world, we actually quickly introduce you after your data exploration and data loading and transforming. We quickly introduce you to advanced statistical methods like machine learning, and with supervised and unsupervised machine learning, and comparing different supervised classification algorithm, and etc. So this is in BioML course in Python, which and also available in R, which compares different machine learning methods for the same or similar data, so that you can clearly get an understanding of how same or similar analysis methods interpret the pattern that is that you can find, or that is available in the data, that is present in the data. So always you start with load and transform and start exploring the data, and then before going into further analysis methods. So all of these methods will actually take the structured data that I was talking to you about. So this is a tabular output that you can generally get access to from publicly available data set, or if you have next generation sequencing data, you can get to this structured data from running one of those pipelines that we have discussed before, and you will also see one more example of that. So the practical coding assignments in our in the portal, on portal, is designed in such a way that it will help you to understand the different elements of code R from coding Python. For example, this is an example of interactive console that we are going to offer you for both Python and R environment, where you'll have your code, where you'll have the code that you can write on the console, and then the console will and the right portion of the console will be where it is being executed, and interactive output is going to be printed out. And you will also have, if you have codes to prepare, or if you have codes to print out plots, and the plots will also appear below. So this is embedded into each and every one of these coding lessons, so that whatever you have achieved in learning the code section, you can apply it into this practice the code area. So and we also have some more test your ability to fill up the codes, where we modify some of the coding codes, so that you can complete those syntaxes, and we deliberately leave out some parts of the code, so that you can access yourself, or you can try to complete those codes, and learn the code section area. So once you are through with the practice section, where you have completed these codes successfully, and these practice sections are designed in such a way to increase the level of difficulties, and once you are through, you will be given either an option to perform an assignment or complete an assignment, or you'll be given access to Google Collaboratory Notebook or a Jupyter kind of Jupyter Notebook kind of link, where you can get all the codes that are discussed in the lesson and a little bit more other options also. So you can get a feeling for running the code either one by one or in unison from start to end. And you also, since you have the access, you also have the freedom to play with the code, to change the analysis methods, to compare one method with the other one, and etc. So as I mentioned at the start, we don't focus on making you learn programming. We don't focus on making you learn programming, but we let you understand the data science aspect of the programming. So how different strategies of these analysis work together, and how by employing different data processing methods will help you define the hypothesis, will help you refine the hypothesis, and will help you prepare the data for meaningful applications. Most of the time in the data science application method, most of the time spent in this portion and this portion. The data analysis and summary of statistics will take only will take limited amount of your time and limited amount of your coding areas. So go back, I think, yeah. So most of the time you're going to spend in processing, cleaning, and pre-processing the input data such that it fits the analysis requirements of, it fits the statistical requirements of different analysis methods. And data mining and visualization and exploratory analysis perform very important key portion in refining your hypothesis. You look for pattern. If that pattern is present, then you refine your hypothesis. Or if some of the initial early exploratory visualization provides you with a different different idea or a different view, so you expand on that view, you go in that direction. That's the power of data science. We don't start with the fixed hypothesis. We explore the data for available hypothesis, and then we keep refining the hypothesis so that our data can explore the relationships in the features or in the genes or in different aspects of different portions of the data to reject or accept or to evaluate the hypothesis. So I have a question where, before going next, when using code written by someone else, there are concerns about copyright issues and whether it is necessary to get coders' permission to copyright. So if you are getting the code, or if you're downloading the code from the coder's computer without their knowledge, then of course you have to get permission. But there are codes which are left for you to explore in the freely available resources like GitHub or in Google Collaboratory. So in these portions, you can just mention modified from this code, so that you are being a good person and then acknowledging from where the code source is being taken from. And most of the publication nowadays require the authors to give link to their code so that people can reproduce their results. So once they give permission, or once they upload the code in downloadable public resources and repositories like Google Collaboratory, Google Colabs and in GitHub, then it is free for you to take, provided that you acknowledge them accordingly. So whenever you want to take codes written by somebody, please acknowledge them. This is a code that is taken from this link, and it is modified to suit this specific this specific analysis method. Okay. So the difference between, for example, choosing a specific omics data analysis track and choosing, okay, I went back again, sorry. Choosing specific omics data analysis track and program and choosing data science programming, data science track is that in the data science track, you will get, you will have experience in dealing with the structured data that is generated from variety of different omics data analysis. You can get either transcriptomics data, you can get a list of variants in the genomics data, and you can get abundance table from the metagenomics data. Each one of these structured data that you can get your hands on requires different concoction or different combinations of programming methods, and so that you can explore the variety of patterns that is associated in the data, that is buried deep in the data. So this can be as simple as, the analysis can be as simple as performing a t-test in Excel, or as complex as performing hierarchical clustering or unsupervised clustering, and then followed by supervised classification for different one of these clusters. So it can go from very simple methods to even complex and complicated methods. So the next program we have in our hand is focused on transcriptomics data analysis for chronic diseases. And in this program, we will discuss various aspects of transcriptomics data analysis, which is also called as RNA-seq data analysis, and how this specific field of bioinformatics has evolved and continue to evolve to help us understand the disease biology or systems biology from the disease biology from the systems biology aspect. So the technique and the analysis methods and the bioinformatics analysis for understanding the patterns within the data and the comparative analysis of this data has evolved and has been refined over the years. Now it is at a point where all of these data analysis can be run without or with minimal user intervention. And it is now regularly used by many scientists and is being performed, used for in-depth analysis of gene expression data and isoform expression data to study the transcriptomic landscape of the diseased state or altered state, etc. So, so we know that the genomics data has provided, genomics and transcriptomics data has provided new ways and new options and new dimensions to link targets to disease and how to validate these targets. The result of this genomics data revolution is a potential for a new era of drug discovery, and transcriptomics is at the right at the pedestal for those drug discovery of the drugs that control disease through regulating gene expression, for example, or by mimicking the mechanisms by which our body exerts control over a range of normal and disease functions. So the underlying data that is required for performing a transcriptomics data analysis, as you would have guessed, comes from next generation sequencing. And let's talk about NGS and what it represents in transcriptomics data compared with the other techniques, and also get a brief introduction to how traditionally mRNA abundance or gene expression abundance has been measured quickly. So, so as a method of measuring RNA translation, transcriptomics studies actually help us to detect mRNA expression inside the cell. And this mRNA expression will act as an accurate alternative to or accurate proxy to the conditions of the cell that it represents. So this conditions and this phenotypic, this transcriptomics landscape or expression landscape can help us understand, for example, the disease biology or the development of resistance for a particular treatment, or generally the difference in the molecular pathways and difference in the biological function that one set of tissues establish, or one set of tissues exert over, compared to the other set of tissues. So at the forefront or at the basic level, we have to measure the mRNA level or the amount of mRNA present in the cells, present in the tissues. So how do we do that? Traditionally, this has been done using microarray technologies. More about this and comparing and contrasting microarrays and NGS are detailed in this program. But with each iterations or each development in technology involved in measuring the mRNA abundance, we get more and more data or more and more information regarding the cell or regarding the phenotype or disease biology of the cell. So in the end, what we would like to have is something like this, where the genes and the isoforms of the genes will actually represent the difference in the type of samples or difference in the phenotype of the samples that it is taken from. So depending upon your analysis methods, depending upon your type of question, you can either go for traditionally understanding or analyzing the gene expression landscape, or you can go for isoform expression landscape, where you will get your hands-on or where you will get information about the alternative splice forms of the genes that you are analyzing here. So this, as we said, this will extending your analysis method to include isoforms will actually help you understand or help you identify some unannotated exons or novel transcripts which have not been characterized before. So that can be very crucial in understanding the disease biology. So these patterns and gene expression patterns and transcriptomic landscape can help you identify or help you understand the biological markers or biomarkers for which is always used for used in the clinical data diagnostics and clinical therapy selection of different therapies and improvement of clinical diagnostics. So more and more of these data and more new discoveries based on these expression patterns and isoform expression patterns will help us understand these complex diseases at the subcellular level, also from the target identification for the new or repurposing of existing drugs or novel drug design. So this is Transcriptomics program, and this program includes 6 or 7 interactive sessions. We will cover all these topics within this interactive session. In addition to this, you will get a practical hands-on where you will understand the logic behind setting up this transcriptomics data analysis, which we will discuss in detail. I will rush a little bit quickly here, and in the end of this analysis, you will get a list of output files which you can either explore them interactively or download them to use it for your report or use it in your analysis. So in addition to identifying differentially expressed genes, which is the aim of performing transcriptomics data analysis, you can also understand how different pathways are enriched or suppressed because of the genes that are differentially expressed in the conditions that we are analyzing or comparing. So the basic, this I will skip. And the outputs are, you will get a myriad or variety of outputs, which will include tables, which will include plots, and which will also include visual output, interactive visual output. So all of this will help you understand the biological phenomena or clinical condition of the disease. So as I mentioned, this structured data can be used for further data science based analysis, and this we already discussed, so we will skip. So you also get an introduction to single cell transcriptomics, which can help us understand all the messenger RNAs that are present in single cell resolution. So it is not like bulk cell profiling, which we have been discussing so far, where the sequence libraries are created for an averaged mRNA expression quantification for a different for a tissue. Here you will get the tissue composition at the single cell resolution. Let's quickly skip through a couple of these parts. We have one more program to cover. So this actually tells the power of single cell analysis, pipeline analysis, where you can, for example, here where you can get different cell types that populate a tissue, so that you can understand the heterogeneity behind the tissue that you are analyzing in a much more clear fashion. So this level of resolution you will not obtain or you will not get from bulk cell RNA analysis. Bulk cell RNA analysis and single cell RNA analysis complements the bulk cell RNA understanding of the disease biology in a very upcoming and a novel fashion. So the next program that we are going to discuss is for Multiomics for infectious diseases. So here we will speak about, in this program, we will discuss about the emergence and development of infectious disease research area and how bioinformatics is contributing to this infectious disease research. So we will actually use again the next generation sequencing data and how they can be used and how they can be employed to characterize and understand the infectious diseases, the pathogens, from a data perspective. So so in this program, we will show you examples of how to perform, or we will provide you training on how to perform sequence variation analysis and alignment analysis and how to perform phylogenetic analysis, so that you can understand, for example, where where your SARS variant or SARS strain relate to or lie in the lineage of variants of concern or variants of least concern. And we will also extend this analysis to understand the functionality and the 3D structure of the protein and how a protein structure can be related to its function and why it is important in the connotation of pathogen infectious diseases and pathogens entry into the host. So in the end, you will also get the host side of omics data analysis. We can record what the host, how the host respond to different pathogenic infection, and from understanding the transcriptomic landscape of host response, we can get an idea of what are the different ways the hosts we as a host are trying to counterattack the viral outbreak and virus output. Will the multiomics data teach us to make hierarchical clustering based on DNA sequence analysis for a particular gene or using? Yes, you can perform clustering using particular gene or a set of genes using coding. Yes, that is included in this as well, in this part as well. So, and yeah, okay, perfect. Let me. Can somebody mute your screen? Mute yourself. I think we have some disturbances coming up. So, okay, thank you. So so I wanted to talk to you a little bit more about infectious diseases, how it is different from non-infectious diseases, and why we are to study the differences between infectious diseases. I will not go over much detail, but we say that infections are caused by agents that we call as pathogens, and these are usually virus, viroids, fungi, parasites, and bacteria. And the non-infectious diseases are caused by nutrition deficiency or caused by chronic development of different chronic response to different factors like environment, lifestyle, and it can also come from genetic inheritance and etc. etc. So I want to give you a special overview of how omics data is actually helping in understanding the infectious diseases case. So at the same time, when we are developing a lot of new techniques to study these pathogens under microscope, we also have started developing sequencing technologies. So these two technologies, especially in infectious disease biology, complement each other. So the data that we get and the biology that we study is rich, and it has completely transformed our understanding of infectious diseases. I don't have to give a lot of examples because we are currently in a pandemic era, and Ebola, the epidemic that we encountered in 2014 to 16, excuse me, is one of the first epidemic to be tracked in real time using next generation sequencing data. So for understanding multiomics integration in infectious disease biology, we don't have to go far, but we can easily take the Ebola pandemic as a case study to understand how different strains of Ebola are characterized and how different outbreaks of Ebola are actually contained by analyzing the next generation sequencing data, and how we are repeating the same for SARS-CoV data. All of your variants of concern and variants of least concern and double mutant, triple mutant of these variations of these Delta and Omicron variants are actually employing next generation sequencing data for their characterization. So as a result, as we know that the omics data is now regularly used to study viral genomes, is used to study DNA variations, is used to study pathogenic gene expression or host gene expression data, and host protein abundance and the composition of different properties of like microbial community in the metagenomics data. So all of these different types of data that we have need to be effectively analyzed and need to be mined for different patterns to understand how hosts respond to the pathogenic invasion and how generally pathogen employs different parts of host protein machinery for its own purpose. So in the end of all these, you will understand and you will develop a deep understanding in the processing and analysis methods that are used to integrate genomic, transcriptomic, and metagenomic data so that you can mine in the, we don't have CRISPR also, but if you can find CRISPR data that will complement omics data, nothing stops you from using that, Shiva. So yeah, we give you methods to study relationship based on, for example, local and multiple sequence comparison using phylogenetic analysis, and phylogenetic analysis will give you a new world into the reconstruction of evolutionary patterns for example, for viruses and for different pathogens. We also give you different, for example, chemical and structural variations that is observed in the sequence, amino acid sequence, that are mapped onto the protein from, for example, in the surface protein of SARS, on the surface protein of Ebola, or in the genes of Mycobacterium tuberculosis, and etc. And we also give you ways to understand the immunology, immune response of the host by studying their transcriptomics data, transcriptomics landscape for a pathogenic invasion. So with that, I can take some questions that you are busy with, or you are typing with. And then if you have no further questions, I will pass the stage to Dr. Mohit, who is going to talk to you about research fellowship.
Thank you, Raghav. I think there are a few questions in
in
the chat. So once you can cover those, I'll share my screen.
Sure. So Vishnu writes, I wanted to find out how disrupting a specific gene impacts other genes and also in protein expression in its network. Do you think bioinformatics using R and Python is a right fit for this goal? You can use, yes, I mean, wherever you can find data to analyze and big data to analyze, you can use R and Python. So R and Python are just really tool to analyze the bioinformatics data. I think you should focus more about what is the specific question and what is the logic behind those analysis. So if this analysis involves you to run these run couple of analysis methods by submitting this to online servers, so be it, you run it, but the end result should be you should understand the disease biology, as you mentioned, to understand how a specific gene impacts other genes. You can, for example, I will give you a hint. You should look for hub genes. You should look for how people identify hub genes from protein co-expression network, gene co-expression network. That will give you a hint on how to perform this analysis and what to disrupt in the gene expression landscape or gene expression patterns. And what else do we have at part? Maybe cancer genomic course would be good for you. I think I am interested in neuro-oncology and metabolomic research. Which program would be helpful for me? Okay. Yeah, I mean, since you have oncology, you can use cancer, and most of the cancer genomics we study use transcriptomics data analysis. So I would point you to transcriptomics data analysis program. So that is, with that, you can probably answer your question trying to understand metabolomic research and neuro-oncology area. Giselle actually writes, I'm interested in species, genetic structure, phylogeny, and chemical composition based on structure. Which program will fill the multiomics program or multiomics data analysis program for infectious diseases would perfectly fit your needs, Giselle. So that includes these analysis methods where we are comparing genetic genome sequences, where we are performing phylogenetic tree analysis, where we are also going to understand how mutations in the genome or gene of specific region will affect will or could affect the protein function in the end. So that will fit your needs. And Van der Koe will, I actually answered the question on hierarchical clustering, and we have thank you. I think with that, I can pass the stage to Dr. Mohit, who's been patiently waiting. Over to you, Mohit.
Thank you, Raghav. So can you confirm if my screen is visible?
Yes.
Yeah.
Yes.
All right. Thank you. Hello, everyone. It's really pleasure to meet you all. So today we are meeting as an orientation session for the 2022 LBR and Biomedical and NEPI virtual summer programs. So in these summer programs, as we have been discussing about different topics and different curriculums, I think a great question would be like, what is the right fit and how they are categorized. So a little bit about that. So the Introduction to Bioinformatics program that we discussed first is a beginner level program that has sessions designed to take you through big data and then take you through the omics technologies. You'll learn about genomics, you'll learn about transcriptomics, and you'll have access to a lot of asynchronous material, which will make this a lot easier for you to learn at your own pace, at your own time, and at your own convenience. We will have live sessions for queries, Q&As, and we will also go through some specific case studies that will help you understand about a specific research, may that be genomic research, that how it can be applied. So that's one of the key advantages of OmicsLogic training. It's just not based on, the technology itself, but also the case studies and how it's applied for research. Now, again, the first program that I discussed and the second program, which will be on data analysis, biomedical data analysis using R and Python, is again a beginner level and also an intermediate level training program. So for this R and Python, I mean, we need to understand that where this is applied, and especially in the context of research. So you'll see a lot of publications published, recent publications in Nature, Cell, and Science, and all the good journals that are out there, high impact journals. They use a lot of visualization, which are really compelling to look at. And also at the same time, one can understand that what this experiment is all about and how can you actually visualize a huge amount of information into a small piece of, I would say, art. So this is where R and Python are really, really massively used. And there is a lot of application, hence in bioinformatics and biological research. So it's a great program for beginner level who have not experienced this and have not been using this for their own publications, reports, and all of that is a good fit. Now, the transcriptomics for chronic diseases, I mean, as Raghav said, transcriptomics is a very important topic in today's modern era in biomedical research. So it is the transcriptomics research is again a beginner and also an intermediate level training program, which because all of these training programs have a very basic introduction that allows anyone from any background to actually learn about the topic. But obviously, basic biology, molecular biology is something that we expect you to know a little bit. And then one of the biggest, expectations that we will have if you're a beginner level is to complete the asynchronous coursework, because they are not just like any other courses. They are more like practical tutorials that will help you like to go through a case study, that will help you to understand that how this is connected with the T-Bioinfo server and how can all of this can be conceptualized into a research, into a research project. And that is what I wanted to discuss today in this session. Now, the last level, which is, I would say, an advanced level, because you will have to work on a project. And I think many of you who are thinking about the project has already some ideas in their mind. Maybe it's related to the research that is ongoing or some of the new technologies that you're hearing about, or it could be about the lab that you're working in and you had some specific interests and specific ideas that you wanted to explore. So it's an amazing program, this research fellowship, I would say, because it's like an experience that takes you to use the high-grade training research tools, and then you get to do the hands-on analysis and then work and apply multiomics data or any sort of data that is publicly available to these different research fields. So it does not stop you to work on a very specific research field. You can have any of the research fields that are out there that uses biological data and generate a research idea and work on that idea. But obviously, there is a requirement for you to know how to use the T-Bioinfo platform, you to know how to use R and Python maybe for visualization, you to know also how do you come up with research question. That is one of the biggest roadblocks to work on a research research project. So, I mean, this entire research, OmicsLogic research training is very well structured. And I would say this is very well structured because we have been able to do this in a very short period of time, considering people end up doing PhDs and they end up working for a very long time to get some of the outcomes that we are getting in a very quick time. And that is all because of the platform. That is all because of because we are trying to simplify all of this. Because this is also a complex way of applying and understanding how big data, how this huge amount of information can be summarized and make meaningful information insights that really helps in translational research. So big deal, yeah. So that's why what we are doing is that we have this basic coursework, then we have these tools, then we have the platform, then we have this research specialization areas, and we talked about special case studies that anyone can go through and learn about a specific kind of research. And then the last step is to start working on your own idea, generate results and interpretation, work with a mentor, work with us, work with a team of collaborators or team of experts and collaborators across the world. So this is what the research fellowship training program is all about. And obviously it guarantees some of the outcomes that we have been able to get through this sort of training. That is that people are being able to highlight, showcase, and also get results to a level that can be a preprint, that can be a poster presentation, that can be a full publication. So more about it, to understand that how do we integrate all of this. There are collaborators from our partner universities. There are OmicsLogic research fellows who are already in this community. So we have a community of over 15,000 people who are researchers, who are students, who are faculty, who are from company. And it's really good to have like a public profile on OmicsLogic Learn, because what we are also doing is that connecting research projects with interests in research companies and companies who are startup companies who are in pharma. So we also have a LinkedIn group where I keep on posting, and where we keep on posting about these job opportunities that are associated with a similar kind of training that you are that you're getting from this sort of experience. Now, what we are working on in a regular basis is some of these cutting edge and modern research areas where big data is used, where biomedical information is used, where clinical information is used, where even lifestyle information is used. So integration of all this data, and for any sort of personalized care, is precision medicine. So there are a lot of projects that we have prepared and worked with many students that specifically discussed about a certain problem. So when it is about precision medicine, then it becomes like really about individuals. So if you think about in the context of an individual, just think about the amount of opportunity that this field is going to produce. Because right now it's like one drug maybe fits a large number of population. Maybe one drug is looked at and given to a entire country. But what we have seen and what we have understood, that due to these subtle changes that we discussed and understood while we are learning about transcriptomics, because we are comparing all of the sequencing and all of the reads that we get for gene expression, and then we are looking at the genomics data, and then we are looking at the epigenomics data, and then the environmental factors that we are understanding from metagenomic data. So it's really now opening up all of those correlations that we were not able to see and we were just guessing. So because of that, there's a huge opportunity in population health, and there's a lot of application in biotechnology and agriculture, and there's this new field called spaceomics. So I can keep on like talking about this for hours and hours. So there's not enough, and there's so much is going on. So these are some of the opportunities, and also like the projects that you saw there are some of the example projects that we have put together that helps anyone to understand that how this sort of research has been conducted, because they are taken from peer-reviewed publications, and as well as they are taken from data sets that are available on Geo, SRA, all the public repositories that are hosted on the amazing NCBI portal. So about the research, the way we are trying to addressing a research, it's more data-driven research. So when we are doing a hypothesis-based research, we have some sort of idea about how this hypothesis of ours will be like. Now, when we are looking at this from a very different prospect, when we are looking at the data first and understanding that what this data can answer. So that's data-driven research. And there is so much of data which is available out there. There is a lot of opportunity for research. So identifying gaps and finding those unanswered questions, defining your question. These are some of the things that we are helping as a mentor to the research community, to the participants who are joining the research fellowship program. And then there is a strict structure that enables us to track, enables us to understand that how idea can be structured into a research project, can be structured into a report, can be structured into a publication. So all of this work goes on when one person joins the research fellowship and go through this entire process of introduction, evaluation, practice, and then communication is one of the biggest key to having a successful training out of this. So all those who have been communicating and coming to the meetings and having this profile that actually showcases, like towards the end when you're completing your research, your project is also highlighted in your profile, and that enables any of the employers, any of the collaborators to know more about your research and reach out to you for the same. So it's very simple to upload this. So we have a mechanism that enables any of the users to upload their own project once they're completed their project. And obviously there's a tracking system that enables us to understand that if you have reached a level where you can start a project or not. So the concept is to earn 10,000 points, and 10,000 points can be easily earned when you start finishing the course and you start running pipelines on Bioinformatics platform. And that will happen when we start the training programs. And before that, our objective is for you to go through the basic coursework that is available for free. And then we will be able to, quickly go through these different topics that we are talking about, exciting topics that we are talking about, and you to make the most out of this training, out of this opportunity. So some of the examples that we've been working, and some of these students have been able to publish, able to complete these projects that involves, working on different of these research areas, including spaceomics, and its application, and seeing how it affects astronauts' health, and what are the different ways we can improve. And then there are very specific research which is related to precision medicine, precision oncology, and very mechanistic related research. So there is a lot of research ideas that we can work with and working with already. And there are already available projects for those who do not have a research idea, but would want to work on a research problem. That sort of opportunity is also there because of these collaborative projects. So from different levels, so we have high school students who are really inquisitive, are into this, have able to finish. Then we have postdoctoral fellows who are using this for some clinical study. Then we have PhD students and undergraduate and master students as well, completing and finishing these coursework and the research project in a very short period of time. So, some of the recognition that people have get, obviously, not just to go for a better university and better degree and all of that, but also to win prestigious prizes, science fairs, and also to be able to show this in our Omics Research Symposium that we do every 2 years, and where we meet with all the experts in the industry, discuss about the recent advancements, and give an opportunity to our research fellows, a platform to our research fellows to showcase their research and win prizes. So that's all about the research part and how we are doing this research fellowship program, and along with the other training programs, which would make sense because once you know about some of the specific training, you can easily apply this to a research project, which is also an option. And this is mostly available throughout the year, but this is like a unique opportunity to work with us within this scope of this program. And also because it's available at we are also providing a 50% scholarships for those who are interested. So a great opportunity for the summer for all of you. And I hope that many of you would join and start working with us in these specific areas of research, utilizing public repositories and public data and our own guidance and the platform and the community. So that is all that I wanted to add to this presentation. And we are very sorry if we have extended this time and we've been talking for a very long time. So I would now like to pass this stage to Sonalika to help all of you with the registration process and understand that how you can start with the utilizing the resources that we just discussed. Thank you.
Thank you so much, Dr. Mohit, for explaining about the research fellowship program. Now, I would like to take you all through the registration process for all of these programs that Dr. Raghavendran explained and also Dr. Mohit. So before I get on to the registration part, I'll quickly go through the portal and help you all how to navigate and pick the resources that you are interested to go through. So first of all, Japs in the beginning had mentioned that you all must have an account on the OmicsLogic Learn portal. So for those who have not yet created their account and joined midway or late, the link is in the chat box. Please refer to this link and have an account for yourself so that you have the access to all of these wonderful resources that we discussed about today. And now moving forward, I'll just quickly go through the profile section. It's very important to have a complete profile. As Dr. Mohit mentioned, that we have various opportunities for those who complete the training, the OmicsLogic training successfully. That's why it's very important for you to have a complete profile, which includes a profile picture. Your name should be in title case, and you should add a brief bio about yourself. That would be about your educational or research background, your research interests. This really helps the mentors know about your progress, your interest, your field, and also help the recruiters pick the best profile. And with this, I'll just go to the courses tab now and explain how you can pick the resources that we discussed about today. On this portal, you have various tags on the right side. So if you're interested to look at the various courses that OmicsLogic provides, just click on the courses tab and you'll get a list of courses, which includes a course on transcriptomics, genomics, metagenomics, epigenomics, data science, machine learning, and you can pick any of these courses. And then you have various example projects as well that we discussed about, from which you can learn about the application of these various different omics data types and understand how you can answer a research question in hand. I think I need to. All right. Now with this, I'll just quickly jump into the registration process steps. I think I need to relog in. Yeah. Okay, so I'll take up the very first program, that is biomedical data analysis in R and Python. Now, these programs which we are discussing about today is specifically for LPRN and NEPI. That's what all of you think. But yes, these programs can also be taken up by people who are not from LPRN and NEPI institutions. And that is why we have this orientation session for all of you who are interested to join these programs. And I'll just quickly go through these registration pages and tell you how you can become a part of these programs. So this is the very first program that I want to discuss about, that is biomedical data analysis in R and Python. I'll just quickly put the link in the chat box to this program page. All right. So once you are on this page, you'll find all the details that are important for this program. First of all, when the program begins, it's 15th of June, and it's a 3 months program that would end on 15th of September, but you can join it asynchronously for a 1 month duration as well. And on this program page, you will find the information about the program's schedule. That is, what all sessions are we going to have? On which all dates are we going to have these sessions? What are the topics that will be covered? And what are the associated resources that you need to go through and complete as a part of a particular session? And how is this program page helpful? Yes, it does give you the information about each and every session, but it will also help you go through the session recordings. So for each and every session that is listed here, we are going to have session recordings, mentor recorded sessions, and I'll be uploading these recordings on the program page on the date of the program, which you can go through. And also you are supposed to first go through the session recording, second, go through these associated resources or these coding lessons that are a part of this program page. Now, when you go through these session recordings and also practice these coding lessons, you will definitely encounter some questions or you would like to talk to the mentor out of curiosity. So we have groups on Skype and also Slack channels which you can join and anytime clarify your question from the mentor. And also we'll be having some live sessions in between these asynchronous sessions where you can meet the mentor and take your questions. Now, how do you register for this program? So if you scroll down, you're going to find 3 different levels for this program. Giselle, I'll just get back to your question. I'll just explain the steps to register first and then I'll get back to your question. All right, so we have 3 different subscription levels. The first is beginner, then we have an intermediate level, and then we have an advanced level. So for all of these different levels, the resources are going to be the same. Yes, the resources, which includes the access to OmicsLogic, resources, which are the coursework, the session recordings, the associated resources, everything is going to remain the same. The difference lies in the time duration for which you are a part of the program and also the access to mentor guidance. Now, in the beginners level, you will be able to have such sessions like the ones we have today, group sessions where we have discussions, you can take your questions, ask your queries, resolve them. In the intermediate session, you can schedule one-on-one meeting with the mentor and clarify your questions. And in the advanced level, you will get some extra time period to work on a research proposal and then finally get started with a research project as an outcome of this training. All right, so that is the difference between beginner, intermediate, and advanced level. The 3 subscription levels and their differences remains the same for all of these programs that I'm going to discuss about today. And there's just a difference in the costing of these programs. So how you can take this particular subscription, just click on the select option button and you'll be redirected to the payment gateway. And that way you can enroll for the program. And those who are opting for scholarships, now I'll take question that Giselle has asked in the chat box. Now, for those who are opting for a scholarship, you need to reach out to us at marketing@omicslogic.com and we'll be able to help you out with your enrollment with the scholarship. Before you reach out to marketing@omicslogic.com, please make sure that you fill out the scholarship form. So I'll quickly give the scholarship form as well in the chat. Yes, so this is the scholarship form. Please fill out the form first and then reach out to marketing@omicslogic.com for the specific program that you are interested to enroll in with the scholarship. And we'll help you get started with your training. After the biomedical data analysis program, let's talk about introduction to bioinformatics training program. Now, again, this particular program page will help you look at various details. That is when this program begins, what is the schedule of this program, all the session details have been mentioned here. That includes the topics, the associated resources, and also you'll have the recordings on the dates of each and every session. And these resources are actually lessons of various courses on the portal itself. And also each and every session will be supplemented with some extra material and also additional videos by our guest speakers and sessions that you'll be getting over an email. And to enroll for this particular program, the process is the same. Scroll down, I'll just give the link in the chat once again. Scroll on this page and you will find these 3 different subscription levels that I explained, and you can pick the one that works the best for you. So this is about introduction to bioinformatics program. If anyone has any question, please put them in the chat box and I can pick it up. All right. So the third program that we wanted to discuss was about infectious diseases, and the link to this particular program is in the chat. So to enroll yourself for the infectious diseases program, you need to visit the program page, and the dates have been mentioned. And please don't bother yourself by looking at this detail for LBRN and NAPI students. As I've mentioned earlier, also the non-LBRN and NAPI students, researchers and faculty can enroll in these programs. The differences lies in the registration steps, and that's what we have been discussing till now. All right, so for infectious diseases, we have the syllabus mentioned here again. I'm not going through each and every session here for each and every program that we are going through because it will be too lengthy. So you have the link, you can go through these session details yourself. And if you have any question, please feel free to reach out to us and we'll clarify the same for you. All right, and to register for this particular program, you can pick up the subscription level that works the best for you. And you have the scholarship form. If you would like to fill it out and get scholarship for the program, you can do the same, and you have the email address as well. And also, I would like to mention here that on each and every program page, you will find a program course and project library. Now, what is this? As a part of this program, you will also get the access to these various resources, which includes, for example, if we talk about infectious diseases program, then along with this program, you're going to get an access to these courses and example projects that I mentioned here. And also you're supposed to go through these resources as a part of the project. So that was about infectious diseases. And now let's go to our next program, which is Transcriptomics for chronic diseases. Till the page loads, let me look at the, thank you so much, Dr. Raghav, for putting the link in the chat. All right, so for this particular program, you again have the dates mentioned. Now, I would like to specifically answer one question that I usually get from the participants. They sometimes ask me that if they're not, let's say, available from 15 June and they're busy till, let's say, first week of July or August, and they would like to enroll then. So can they? So the answer is yes. You can enroll any time between 15 June till 30th September. It definitely has to be a 1-month training program for you then that you can follow asynchronously. So you can enroll in these programs according to your own schedule. And because we have these resources asynchronous, that is, you can go through these session recordings at your own pace, and that is why you can take up the resources, the program, even if you have a busy schedule. You can go through these resources at your own pace, so you can definitely enroll and get started with your training process. And again, we have the sessions schedule, the resources mentioned, and also the topics mentioned for each and every session for this particular program. And then we have the subscription levels mentioned here for the program, which you can pick, which works best for you. So the link is in the chat box and you can enroll in the program. Now, last but not the least, let's discuss about the research fellowship program. So after training program, project sessions will start, or is it simultaneous? If you want to get enrolled in the research fellowship program, let's get started with your research project after a training program, then the first thing that I would suggest to you is that an advanced level works the best for you, where you can actually work with the mentor on designing your research proposal and then get started with your research project, right? So the project sessions, as in you are saying, it really varies from subscription to subscription level for each and every participant. And if you're interested to take it up, then yes, it will start after the training program, not simultaneous, because it's important for you to actually learn about the concepts, run the pipelines, and see how everything has to be like how your data has to be processed, how you run the pipelines, how you get the outputs, how you interpret your outputs, right? So you need to get a training first, and only then you can work on a research project. And if you are already trained with a specific type of data analysis that you would like to work on in your research project, then yes, you can work simultaneously, or simply start with the research fellowship program where you work on your research project. Yes, exactly. But when you talk about the training program, I'm talking about just the training program. And if you want to be a part of the research fellowship program, so it will be like a training program first, and then 3 months or 6 months of the research fellowship program. It would be that means. I hope I was able to answer your question. And if you still have any question or confusion, you can definitely unmute yourself also and take up your question. All right, I think I was able to answer your question. Next, we have a question by Mukesh. Dr. Raghav, can you take the first part of Mukesh's question where he's asking about is biomedical data analysis R and Python beneficial who have to work on ML data science or only for researcher? I'll take up the payment question. For Mukesh, I think you're the best person to answer this question. That's why I'm referring it to you.
Okay, biomedical data analysis R and Python beneficial who have to work on machine learning data science or only for researcher? Now, what is the contrast here? I mean, yeah, if you want to work on, if you want to understand how machine learning and various algorithms of machine learning is employed for big data bioinformatics analysis, then you should use biomedical data analysis R and Python. I mean, specifically, we have named it in such a way that the focus is on data analysis part, is on data science part, not on the biology part. I mean, the biology part comes after you have completed the analysis and after you have gotten the results, and how do you relate and how do you interpret the results with the phenotypical changes or with the disease biology or with the systems annotation and etc. So more and more focus is going to be about applying different and various methods, applying various ML method, machine learning methods that is suited for different data and different question. So it is also suitable for a researcher who wants to get an introduction or who wants to understand how machine learning methods are employed for bioinformatics data analysis using big data. And if we also had students who were well versed in machine learning data analysis only to and have not worked with the biological data. So the focus is going to be using biological data or big data, big genomics data, what all you can learn and what are the challenges and how to overcome them for employing different machine learning algorithms or big omics data. So that is going to be the focus and forefront of this. So I think I partly answered your query. If you want to learn all the nuts and bolts of understanding or of nuts and bolts of using machine learning data, then you have to take biomedical data analysis course.
Dr. Raghav, and thank you, Sparsh, for answering Parth's question. I think, Sparsh, you should reach out to Parth maybe, and Sparsh is the best person who can help you enroll in the program and get started and answer all your questions. And Mukesh, to answer your last part where you were actually asking about the payment method in INR or others. So I would like to specify here that you can definitely proceed with the payments on the portal by choosing the select option button. But if anyhow you face any issues, then yes, we do have alternate payment options as well. Yes, in INR, you can use various different platforms to proceed with the same. And to do so, Sparsh is the best person. She'll help you out with it. Mukesh, please share your email address so that we can send you out all these details in more detail. And also, Pat, it would be great if you could share your email address as well so that we can reach out to you with each and every detail. Van, with a beginner subscription, it's not possible for you to make a project in the end, because you'll just have an access for a month's duration where you will be able to just complete the training part. All right. So if you pick up the beginner subscription level, let's say, you can complete your training first and then get started with your research project by enrolling yourself with the research fellowship program. That would be the best case for you. And if you pick up the advanced subscription, then yes, you can work on your research proposal by actually, because you will get some extra days, that would be around 15 to 20 days where you can actually work on your research proposal and get started with your research work. So yes, that's possible with an advanced subscription plan. Thank you so much, Sparsh, for sharing the information. Sparsh has shared the contact number for Indian citizens who can reach out to us by calling out on this number. And if you are not in India and you want to connect with us via phone, you can maybe use WhatsApp and reach out to us. Anyways, email is the best option that we have shared in the chat, marketing@omicslogic.com. And please make sure that you have this email address added to your address book so none of the emails land in promotions box. Okay, so now I would finally like to discuss about the research fellowship program that we have been talking about, and it really seems that a lot of you are interested to work on a research project. So how do you get to the research project page? Here in the menu bar, you can see this research option here. If you click on the research option, you'll be redirected to the research fellowship page where you can enroll yourself for 3 months or 6 months. Now, if you go to this research fellowship page and click on the Get Started button here, you'll find two options as we have discussed earlier, 3 months and 6 months. You have two levels here, beginner and advanced. Now, in the beginner level, you will get the access to educational cloud pipelines, and in the advanced level, you will get an access to the research cloud pipelines, where you can get an access to an FTP account as well if you have data generated at your own lab. So that is the main difference between the beginner and the advanced subscription level. You can pick the level which suits you the best and proceed further. Now, again, if you want to enroll in the research fellowship program, as Dr. Mohit mentioned, that we have been providing some scholarships to interested participants, you can reach out to us on our email address and also fill out the scholarship form. And our team will review your application and get back to you with your scholarship, and then you can get started. And if you want to enroll for the 6 months, you simply click on this button here and you'll get the options for 6 months. So that was it about all the registration steps for various programs that we discussed and also the research fellowship program. And now I would like to mention here one more thing. Today we discussed the registration steps for students, researchers, and faculties who are not from LBRN or NEPI. Now, if you are from LBRN and NEPI and you want to enroll in the program, for NEPI, you need to simply email us at marketing@omicslogic.com with the program that you're interested to enroll in. And also you need to make sure that you have signed up on the portal and you have an updated profile. Please let us know when you have covered these steps and email us and we'll get you started with your training under NEPI. And if you are from LBRN, you need to register on REDCap. If you are from LBRN, I'll repeat, you need to register on REDCap, and we'll get your registration via REDCap and get you started with the training program. Yes, Giselle, the course, the training program that we discussed, and the research project are two separate phases, not one, but there are two separate phases. In the training program, our focus is just to get you trained and get a good hands-on with our example projects to get you up to work on your own research question. And to get started with your research work, you then need to get you need to enroll for the research fellowship program. Yes, Giselle, that would have another batch of fees that you need to proceed with. And again, scholarship is applicable on both of these, on the training programs and also the research fellowship program. So you'll have it covered.